Now, let's talk about mutation. This is one of these things where I feel like I should put in one of those dun 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 scary music sound effects. Mutation actually, well, it can be scary. It can be perfectly fine. Every now and then, it's occasionally really great. Granted, you're not going to become a mutant and be able to teleport from one spot to another or suddenly grow wings, but occasionally we do get beneficial new mutations that allow us to do things like eat milk and cheese after we're weaned from our parents. So, what is a mutation? Why does it matter? One of the ways that we can categorize mutations is into spontaneous and induced. This refers to how we get them. All mutations are changes to the DNA. Spontaneous mutation just happens as the DNA is replicated. Why? Well, DNA polymerase has a really high proofreading activity, but it's not perfect. It's just really, 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 really good. We're talking 1 in a billion bases are wrong. It makes more errors than that, but the vast, vast majority of them get fixed. So you might imagine some medieval monk that could copy a holy book thousands, tens of thousands of times without making a single mistake that they didn't fix. It's pretty impressive, right? But occasional mistakes still do sneak through. Why? In part, it's a trade-off between perfection but inability to actually ever complete the job because you're constantly going back and ripping out pages afraid there might be a tiny error. And hey, you can do it real fast, but you can do it really, really badly. Spontaneous mutations mean that we all have somewhere approximately on the order of 40 differences in our DNA that we got from our moms between that and our mom's DNA. And same for our dad. So we're going to be just a teensy bit different from our parents. Not a ton, but just a teensy little bit. Induced mutations are what we mean when something has increased the error rate. It's not just DNA polymerase made an error during replication, it's we've got a higher than background level. In general, induced mutation, we're talking about mutagens. That's just a term for things that generate mutations. There are chemicals that will do this. Radiation, high energy radiation will also do this. So microwaves, no, they won't do a darn thing other than heat up water. Light, not a problem. UV though, UV, that can damage your DNA. And the vast majority of the time, your cells will successfully repair that damage to the DNA, but occasionally they won't. And more high energy radiation can cause bigger effects, more damage, make it harder for your cells to repair themselves. The good news with all of this, though, is that the vast majority of these mutations are caught and they are fixed. And even in the cases where they get through, we have 2 copies of every single gene, with some exceptions in people who are chromosomally male, which means that even if one of your copies gets broken, doesn't work anymore, the other copy, still totally fine, no problemo. So, that's great news, except when it isn't. One problem that we do tend to run into, and I'm sure you're all familiar with, is cancer. Cancer is what happens when we get mutations, either spontaneous or induced, in certain genes that keep our cells from dividing out of control. A lot of times, if stuff goes wrong in a cell, say it's missing both copies of an essential gene, certainly in that case it'll die. But even there's a lot of cases of fail-safes where if a cell just isn't doing quite right, it'll just go ahead and die. We've got lots of skin cells. We've got lots of muscle cells. We've got lots of brain cells. Losing one here and there, since we're multicellular, not a big deal. But occasionally those fail-safes themselves get mutated. And now that cell is able to escape and keep dividing and keep dividing and keep dividing. And remember that spontaneous mutations happen during DNA replication. So the more that cell divides, the more mutations it's likely to accumulate. And the more likely it is to gain a mutation that's going to allow it to do things like invade tissues, break off and invade other parts of the body, be able to escape chemotherapy. This is one of those reasons where a lot of times when a benign cancerous growth is found, even though it's currently benign and it's not causing any problems, in many cases that'll still get surgically removed because it's dividing faster than it should. And every cell in there that divides when it shouldn't be dividing is potentially getting closer to actually becoming something malignant and really nasty. Okay, so that's sort of the big picture of mutations. Now let's actually talk about what these changes in the DNA do. Now this particular image is only concerned with mutations that affect the protein. These are only mutations that are in the genes. Why is this the thing we care about the most? Because these are the ones that are most likely to affect traits. But just as an aside, do note, you can have a mutation. Typically this will occur in or around the promoter for a gene, which is going to change how much of the protein gets made, or maybe under what conditions the protein gets made. So if you make lots of a protein that's important for cell growth, or you turn off a protein that usually puts the brakes on uncontrolled cell division, these are also ways that you might potentially increase the likelihood that a cell becomes cancerous. But okay, we're talking about a gene that codes for a protein. So in this case, the example is the DNA is TTC, meaning the mRNA is AAG, and that codes for lysine. The lysine tRNA is going to attach to that, bring that into the ribosome, bring in a lysine. We're cool. One way that mutations can happen, and a way that, frankly, we just generally don't worry about, is what's called silent mutations. Why silent? There are 20 amino acids. There are 64 potential codons. So, there are a lot of amino acids that have more than one codon that codes for them. In the case of lysine, if the DNA is TTT, giving us the codon AAA, that still codes for lysine. It doesn't actually matter. The protein that we make is the same, which means it's going to function the same, which means our traits are going to be the same. No biggie. However, there's a couple that can be a bit more worrisome. A nonsense mutation, as in it does not make sense, or to be more clear about it, it does not make an amino acid, is a mutation that changes the codon from an amino acid codon to one of the three stop codons. One of the three codons that tells the ribosome, you are done making a protein now, you should cut this thing free and let it float off and do its job. What that means is that when this first T is changed to an A, which means the first A in the mRNA is changed to a U, that gives us a stop codon. And not only does that delete that one lysine not only does that delete the one lysine amino acid out of the protein, it also means that every amino acid after that never gets assembled onto it. You've just truncated the protein. You've made it some amount shorter. Maybe a little, maybe a lot. Nonsense mutations almost always affect protein function, and generally affect it by making the protein completely non-functional because it's frequently missing important parts of itself. However, nonsense mutations aren't particularly common because there's only 3 stop codons, and not all codons are going to be able to go from themselves to a stop. So, more common way that we can mess things up is a missense mutation. It is making sense, that is, it is making an amino acid, but it's a mistake. And these can be further categorized as conservative or non-conservative, if we really want to get into it. By which I mean, some amino acids are relatively similar. Lysine and arginine, they're actually not that different chemically speaking. I mean, they're different. This difference could be enough to change protein function, but then again, maybe it doesn't. A non-conservative one, threonine. Threonine is chemically quite distinct from lysine, and it's even different in size. If you look at the purple boxes versus that green box for the threonine, it just looks different. And so this is likely to cause a change in the protein function. Not all missense mutations are going to change protein function, but they can and often do. So again, DNA contains instructions for proteins. These proteins control our function and thus our traits. If you change the DNA, if you change a gene, this means that you change the protein. This uppercase delta is essentially a shorthand that we use for changing something, and you therefore change the traits, or rather the trait in many cases, that this particular gene affects. It could be something small and subtle, like ability to digest something or the way that some cells react to stressors. It could be something really big and obvious, like, for example, albinism, the complete inability to make skin pigment. It just depends. What gene did you change, and in what way did you change it? So what causes genetic diseases? They're caused by mutations in the DNA. That's the long and the short of it. Definition of a genetic disease? Well, it's a disease, so something's gone wrong, and it's genetic, it's in the DNA. Some of these are going to happen spontaneously. There's always a very low rate, but a non-zero rate, of birth defects, where when we finally figure out what mutation is responsible for it, it's not found in either parent. And so this was something that happened spontaneously in either the egg-producing cells of the mom or the sperm-producing cells of the dad, resulting in this novel genetic disease in the child. Environmental factors will also change DNA. UV light, smoking, these are big risks for it. Now note, the likelihood of you giving your child a new genetic disease is extremely small. And so, since you're not generally irradiating your gonads or blowing smoke on them, it's not a huge deal, but it is potentially there. Inherited genetic diseases are the ones where those get passed on in the gametes. One of the activities you're going to be doing this week is actually looking at some genetic diseases, and I would encourage you to follow the link to the genetics home reference. This is a NCBI website. And so there's a huge variety of these that affect essentially every organ system in the body in all sorts of interesting but sometimes also tragic ways. I would encourage you to look into this. Genetic diseases are really fascinating because they elucidate what those genes normally do, what their normal function is, how they normally interact with other things. But a lot of these, like sickle cell anemia, hemophilia, porphyria, SCID, These are also really tragic diseases that kill people young, and inherited forms of Alzheimer's are definitely not good for anybody.